Good morning. I'm Merlin Shiba, Associate Professor, Department of Community Health Nursing, the Bharat Institute of Higher Education and Research, Chennai. Today's topic is about gene therapy from the topic genetics. So, what is gene therapy? The approach of treating disease by either modifying the expression of an individual genes or a correction of an abnormal genes. So, this can be accomplished by replacing a mutated gene that causes disease with a healthy copy of a gene, inactivating or knocking out a mutated gene that is functioning improperly, and introducing a new gene into the body to help to fight the disease. Gene therapy is a novel treatment method which utilizes the genes or short oligonucleotide sequence as therapeutic molecules instead of a conventional drug components. So this technique is widely being used to treat those defective genes which contribute to the disease development. It involves the introduction of one or more foreign genes into an organism to treat hereditary or acquired genetic defects. So if a patient is having any genetic disorders, so we can able to treat with this gene therapy by introducing artificial gene to the body, and we can able to correct that that is wrongly formed genes. The disease is treated with a minimal toxicity, but the expression of inserted DNA by the cell machinery. In 1919, the Federal Association for the first time approved a gene therapy experiment on the adenosine deaminase with severe combined immunodeficiency disease in the United States after the treatment of Ashanti disease. The many diseases such as adenosine deaminase and severe combined immunodeficiency, its X-linked disease, liver congenital amaurosis, a retinal disease, Parkinsonian disease, multiple myeloma, chronic and acute lymphocytic leukemia, adrenoleukodystrophy has reporters for sexual clinical trials. So all these diseases being treated, we can able to treat it with this gene therapy. But these are still not approved by the Federal Association. But some other diseases on which gene therapies based research is still going on, like hemophilias and some other diseases like cystic fibrosis, and some other cancers is also being treating with gene therapy, but still it has been on the process. After 30 years of research and the clinical trials, only one product called Glybera got approval in the month of November 2012, which was made available in the market in the late 2013. It has been ability to cure the lipoprotein lipase deficiency, a very rare disease. And what are the types of gene therapy? There are several approaches for correcting the genes, the most common being the insertions of a normal gene into a specific location within the genome to replace a non-functional genes. Mostly it has been done with introducing a gene to a specific locations where it has been non-functioning genes is being presented, and it classified it as two types. One is somatic gene therapy and germ-line gene therapy. We'll see what is somatic gene therapy. In somatic gene therapy, the somatic cells of a patient are the targeted cells for a foreign gene transfer. In this case, the effect caused by the foreign gene is restricted to the individual patient only and not inherited by the patient's offspring or a late generation. And the germ-line gene therapy, the functional genes which are being integrated into the genome or inserted into the germ cells, that is sperms or the eggs, can be introduced. The targeting of germ cells makes the therapy inheritable. And you can see the difference between the somatic cell gene therapy and the germline gene therapy. First is the somatic cell gene therapies, where the therapeutic genes transferred in the somatic cells, whereas in germline gene therapy, the therapeutic genes transferred in the germ cells. For example, introduction of genes into the bone marrow cells, blood cells, skin cells, or stomatic cell therapy, whereas genes introduced into the eggs and the sperms or comes under the germline gene therapy. And the somatic will not be inherited later generation, but in the germ line, it is heritable and passed on to the later generation. And the somatic cell gene therapy, at present, all researchers directed to correct the genetic defects into the somatic cells. For the safety purpose, ethical and the technical reason, it is not being attempted at present in the germline gene therapy. And here you can see how can we cleanse the genes, that is through the germline therapy. The one-way germline therapy may be done to ensure a man with a genetic disease doesn't pass into its child. That is, it should not pass to its child. So how can we do this? The doctor removes the man's sperm producing cells which contains a defective genes. Next, the healthy genes is added to the each cell to replace the defective ones. And the cells are put into the mouse test. They'll be using with the mouse test, and they mature inside the mouse and start producing the healthy human sperm. And those sperms, once tested, are used to fertilize a woman's egg in a laboratory dish, and the resulting embryos are placed in a woman's womb, and she gives birth to a child whose genes are free from the father disease. So this is a way how they are treating the uh, father's genes that should not be, cause any problem with the child genes. It has been done with the cleaning genes. The methods of gene therapy. So there are two approaches to transfer the genes in a gene therapy. The transfer of genes into the patient cells outside the body is called ex vivo gene therapy. That is being done outside the gene. The transfer of gene directly to the cells inside the body is called in vivo gene therapy. That is happening inside the body. We'll see what is ex vivo gene therapy. This is a mode of gene therapy where the genes are transferred to the cells grown in a culture. Transformed cells are selected, multiplied, and then introduced into the patient. The use of autologous cells avoids the immune system rejections of the introduced cells. The cells are sourced initially from the patient to be treated and grown in culture before being reintroduced into the same individual. This approach can be applied to the tissues like hepatopoietic cells and the skin cells, which can be removed from the body, genetically corrected outside the body, and reintroduced into the patient body, where they become encrafted and survive for a long period of time. The isolated cells with genetic defects from a patient, uh, it will grow the cells in the culture, then they'll be introduced in the therapeutic genes, and they'll select the genetically corrected cells and grow, and then it transplant to the modified cells to the patient. So in this picture, you can able to find how they are doing the ex vivo gene therapy. First, therapeutic genes, that is the genes is inserted into the viral DNA, which is being cultured, that is culture cells are infected with a genetically altered virus. That is, a defect cells are removed from the patients, then the cultured cells are done with a culture. The cells are grown in a culture, whereas the target cells are now genetically altered with a therapeutic gene, Then the cells are reintroduced in the patient body. At last, you can see inside the body, the genetically altered cells are produced, the desired proteins encoded by the therapeutic genes. That is, outside the body, it is being treated. Example for ex vivo therapy is the first gene therapy to correct the deficiency of enzyme adenodeaminase, which is performed on the 4-year-old girl Ashanti Del Silva, was suffering from the severe combined immunodeficiency disease, which is caused due to the defect in the gene coding for ADA, that is adenosine deaminase, and the deoxyadenosine accumulated and destroyed the T lymphocytes, whereas it disrupts the immunity of the child and suffer from the infectious disease and die at the young age. So this is a picture how you can able to rectify how it has been done. In vivo gene therapy, in vivo gene method of gene transfer involves the transfer of cloned genes directly into the tissues of the patient. This is done in case of tissues whose individual cells cannot be cultured in the vitro in a sufficient numbers like brain cells, where reimplantation of the culture cells in the patient is not efficient. The lymphosome and certain viral vectors are employed for this purpose because of lack of any other mode of selection. In case of viral vectors, such type of culture cells were often used when have been infected with a recombinant retrovirus in vitro to produce a modified viral vector regularly. These culture cells will be called as a vector producing cells. The vector producing cells transfer the genes to surrounding disease cells. The efficiency of the gene transfer and the expression determines the success of this approach because of the lack of any way of selections and amplification of cells which take up and express the foreign genes. Examples of in vivo gene therapy mostly takes place in the disease condition called cystic fibrosis. In patient with cystic fibrosis, protein called cystic fibrosis transmembrane regulator is absent due to the gene defects. So in the absence of this cystofibrotic transmembrane regulator, the chloride ions concentrate within the cells and it draws water from surrounding. This leads to the accumulation of sticky mucoses in the respiratory tract and the lungs, where it's treated in vivo replacement of defective genes by adenovirus vector. So you can see here in these pictures the normal human genes flowed into the bacterium, whereas the normal genes inserted into the harmless virus, the virus dripped into the lungs through a thin tube inserted in the nose. Mostly in the case of cystic fibrosis, the mucus secretion which will be become very thickened and it will stop and it will create lot of respiratory problem. So for this they are inserting a tube, whereas the virus will drip into the lungs through a thin tube which is inserted into the nose and it is being sent to the lungs where the virus enter the lung cell nucleus and the lung cell and it is being treated the normal genes DNA sequence inserted into the lung cells DNA by the virus, where you can able to see here the cells now with the normal genes functioning normally. In vivo gene therapy is a direct delivery of the therapeutic gene into the target cells into the patient body, which is carried out by the viral or non-viral vector system. It can be the only possible option in patient where individual cells cannot be cultured in vitro in insufficient numbers. That is, in vivo gene transfer is necessary when cultured cells cannot be replanted in patient effectively. You can see the difference between in vivo and ex vivo gene therapy, which is less invasive, and the ex vivo is more invasive. It is technically very simple, and the ex vivo is technically very complex, and the vectors introduced directly No vectors introduced directly. And in vivo, it's a safety check not possible. And the safety check is possible with the ex vivo. And the disease control over the target cells and the close control is possible with the exo vivo gene therapy. And you can see the what are the target cells for the gene transfers. And here you can see what are the diseases can be a disease and what are the target cells with these diseases. First is the cancer, where the tumor cells and the antigen presenting cells, blood progenitor cells, T cells, fibroblasts and muscle cells are being used in this as a target cell. An inherited monogenic disease, that is lung epithelial cell, macrophages, T cells, blood progenitor cells, hepatocytes, muscle cells, or these are the target cells which is being used in this gene transfer. Infectious disease, that is T cell, blood progenitor cells, antigen presenting cells and the muscle cells. Cardiovascular diseases, that is endothelial cells, muscle cells or the target cells. In the rheumatoid arthritis disease, synovial lining cells are the target diseases cells. If we can see it is a vectors, what are the vectors in gene therapy? To transfer the desired gene into the target cell, a carrier is required. So that is called the vector. Such vehicles of gene delivery is known as vectors. The two main classes are viral vector and non-viral vectors. So you can see how the application of gene therapy in this picture, where you have a lot of vectors and which is being introduced into the gene, and you can see the healthy cells here. First vectors is retrovirus vector system. The recombinant retrovirus has a ability to integrate into the host genome in a stable fashion. That is, can carry a DNA of size that is less than 3.5 kb. The replication defective virus particles target cells it's dividing. Next is the adenovirus with the DNA genome. It is called as a good vectors where the target is non-dividing human cells. For example, the common cold adenovirus. So you can see the gene therapy, how with the help of adenovirus vectors, how it is being used in this picture. Next is an adeno associated with virus vector. It is a human virus that can integrate into the chromosome 19. It is a single-stranded, non-pathogenic small DNA virus. That is adeno-associated virus enters the host cell because becomes a double-stranded and gets integrated into the chromosome. Next is an herpes simplex virus vector. The viruses which have natural tendency to infect a particular type of cells, they infect and persist into the nervous cells. And you have a non-viral vector system. One is called a pure DNA construct and the Lipoplexis. What is pure DNA construct? A direct introduction of pure DNA construct into the target tissue. Next is the efficiency of DNA uptake by cells and expression rather low. Consequently, the large quantities of DNA have to be injected periodically. Next is lipoplexis. The lipid DNA complexes. The DNA construct is surrounded by artificial lipid layer, which is most of it get degraded by the lysosome. And the third one is a DNA molecular conjugate. A commonly used synthetic conjugate is polyl lysine bound to specific target cell receptor. That is, a therapeutic DNA is then made to combine with the conjugate to form a complex. It avoids the lysosomal breakdown of DNA. And the last one is in the non-viral vector system, the human artificial chromosome can carry a large DNA, that is, with one or more therapeutic genes with the regulatory elements. And what are the methods you can do this gene therapy? One is the physical method and the other one is a chemical method. One is in a physical method, you have a gene gun. That is, the employs a high pressure delivery system to shoot tissue with a gold or tungsten particles that are coated with DNA. Next is a micro-injection, the process of using a glass micropipette to insert the microscopic substance into a single living cell, normally performed under a specialized optic microscope setup called the micromanipulator. Next is a chemical method using a detergent mixture, that is a certain charged chemical compounds like a calcium phosphate or mixed with the functional cDNA of desired function. The mixture is introduced near a vicinity of recipient cells. The chemicals disturbs the cell membrane, widens the pore size and allows the cDNA to pass through the cells. Next chemical method is liposuction. It is a technique used to inject genetic material into a cell by means of liposome. Liposomes are artificial phospholipid vesicle used to deliver a variety of molecules including DNA into the cells. Next, the other types of gene therapies are gene augmentation therapy. The most common form of gene therapy is the gene augmentation therapy, where the foreign genes are replaced with the missing or the defective genes, which is normally done in the gene therapy. For example, the replacement of defective P53 gene by a normal one in a liver cancer. Next is a gene inhibition therapy. It is done to block the overproduction of some proteins, that is two types, antigen and antisense therapy. What is antigen? It blocks the transcription using an antigen oligonucleotide. The antisense is to block the translation using an antisense oligonucleotide. What will be the disadvantages of gene therapy? One is long-lasting therapy is not achieved by the gene therapy because it is due to a rapid dividing of cells. Benefits of gene therapy is very short-lived. It is not be a long-lasting therapy. We can't use for the long-lasting therapy this gene therapy. Next, the human response to the transferred gene stimulates a potential risk to the gene therapy. Mostly the immune response will become a very challengeable thing in the gene therapy that will be a disadvantageous. And viruses used as a vector for gene transfer may cause toxicity, immune response, and inflammatory reaction in the host. The disorders caused by the defects in multiple genes cannot be treated effectively with the gene therapy. For example, the multiple genes are being defected. We can't able to treat with the gene therapy. Either one or two becomes defected or missing genes can be treated with gene therapy like in a liver cancer such as like that. But if it is a multiple genes are become defective, it is very difficult for the gene therapy to get success. And what will be the advantages? The gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis, adenosine deaminase, and severe commonly immunodeficiency virus. It is possible to cure for a heart disease, AIDS, and cancer. It gives someone born with a genetic disease a chance to live. It can be used to eradicate diseases for the future generation because the father's genes can be cleansed that it is not going to spread to the children. And some of the ethical issues when we are doing gene therapies are who will access to this therapy? It is interfering with the God's plan. Some people are thinking that it is not according to the God's plan. Should people be allowed to use gene therapy to enhance the basic human traits such as height, intelligence, etc.? It is all right to use the therapy in a prenatal stage of development in babies. And conclusions. So theoretically, the gene therapy is a permanent solution for the genetic diseases, but it has a several complexity at the current stage. It is not accessible to most people due to its huge cost, because we can see it is not, it is going to be a very cost effective procedure to do this gene therapy. Not everybody can be easily access to this gene therapy because of this cost effectiveness. Not the more people cannot affordable with this gene therapy, and this is and most advantages, the gene therapy can give a permanent solution for the genetic diseases. A breakthrough many come anytime, and a day may come when almost every disease will have a gene therapy. And the gene therapy have a potential to revolutionize the practice of medicine. So nowadays, the gene therapy has become a very, very updating technology to in order to treat cancer, AIDS, or even any defective genes or missing genes can be easily changed through gene therapy, but still it has certain disadvantages also. But there are treatment, that is successful rate is also becoming high. I think so far we discussed about the gene therapy and what are the types, exo and in both gene therapy. I hope you got an valuable information from my lecture. So thank you all.